Okay, so we were continuing with the mitochondrial DNA, and now I have taken from the internet source from a PDF book. Well, this is the structure of the mitochondrial DNA genome. It has been organized into certain regions. You can see here. Well, let us reveal these regions. What are they represent? Okay, and I have also not able to mention about this ND6 region. We will reveal them here. Okay, so this is the structure of a human mitochondrial genome, which consists of 16,569 base pair encoding 30 respiratory complex subunits, 2 ribosomal RNA and 22 transfer RNA. The encoded proteins are localized in inner mitochondrial membrane and are some of the core subunits. So we have seen that the human mitochondrial genome consists of this much amount of base pairs, which encodes about 13 respiratory complex subunits. It also encodes 2 ribosomal RNA and 22 tRNA, and the encoded proteins are all localized in the internal mitochondrial membrane, and some of the core subunits of the respiratory complex 1, 3, 4, and 5 has been mentioned here with the help of color identification. Well, you can see all the yellow ones, okay, which represents the subunits from the complex 1 of respiratory complex. Then the purple one that you can see, the green one from complex 4, and the orange one from the complex 5. Okay, so about this mitochondrial structure, DNA structure and organization, you have to name them this ND4, ND4L. Okay, so I have already discussed with you this one. This part is already been discussed by me. Okay, now coming to chloroplast genome. Chloroplast genome is much more greater than the mitochondrial genome, and the size of chloroplast genome is approximately 100 to 200 kilobase pair. About 150 genes are involved within the chloroplast DNA. Like mitochondria, chloroplasts contain their own genetic system, reflecting their evolutionary origin from the photosynthetic bacteria, blue-green algae. It has been proposed that chloroplast also come from the blue-green algae photosynthetic bacteria, according to the endosymbiotic theory. Okay, as they have their own DNA. Chloroplast DNA are circular in nature, that are present in multiple copies per organelle, just like we have seen in mitochondria. Chloroplast DNA are also circular in nature and they are found in multiple copies. Now, these are some about the different organelles, different organisms who have their chloroplast DNA in varying amount. You can see here. So it varies. Okay. Well, the chloroplast genome encodes a variety of proteins that function in photosynthesis, protein folding, and metabolism. So different function played by chloroplast. And the DNA which is found in the chloroplast They encode some proteins. That proteins have various functions related with photosynthesis, related with folding of the proteins and metabolism. Most of the genes that are found within the chloroplast DNA have encoding three different types of rRNA like 23S rRNA, 16S rRNA and 5S rRNA. It also encodes for 27 to 31 different tRNAs, about 40 different ribosomal proteins of chloroplast ribosomes. It also encodes different proteins which are the components for the PS1 and PS2, which are called as photosystem 1 and photosystem 2, which are multi-subunit membrane protein complexes which are involved during photosynthesis. Normally, we all know that this photosystem 1 absorb long wavelengths of light, whereas photosystem 2 absorb short wavelength of light, which are needed for the electron transport for phosphorylation. Okay, so all these are encoded by the chloroplast DNA. Okay, so just go through this. These I not need to mention. Just go for this. The different amount of chloroplast DNA found. This is the main thing that I have mentioned with you. That what are the functions? What are the gene it carries? It carries gene for the three different types of rRNA, different tRNAs, about different chloroplast ribosomes. It also include and the proteins which are related with photosystem 1 and photosystem 2. Okay, actually not all the photosystem 1 and photosystem 2 components I encode. The proteins which are involved for the photosystem 1 and 2 are actually encoded by the chloroplast genes. It also encodes, the chloroplast DNA also encodes the larger subunit of Rubisco, whose name is ribulose 1,5 bisphosphate carboxylase oxygenase. Well, you heard this name during photosynthesis. So Rubisco, its large subunit, is encoded by the cpDNA or chloroplast DNA. Rubisco or ribulose 1,5 bisphosphate carboxylase oxygenase. It is a copper containing enzyme, and this enzyme involved in the first step of carbon fixation. This enzyme is the central enzyme for photosynthesis and probably the most abundant protein which is found on the earth. Right? Rubisco enzyme made up of two protein subunits, large and small. Now this large subunit of Rubisco, name of that large subunit of Rubisco is encoded by a gene called RBCL gene. Small letter RBC, capital L gene. This gene is located within the chloroplast DNA. And this gene is responsible for the small subunit Rubisco are located in the nucleus. So Rubisco is made up of two subunits, large and small. The large subunit is coded by RBCL gene, and this gene is located within the chloroplast genome. Whereas the gene responsible for the small subunit, they are located within the nucleus. Now they will be further imported to the stromal components of the chloroplast through the chloroplast transporter proteins after its expression into proteins. So it means to say the gene responsible for the small subunit of Rubisco, this gene is located within the nucleus. For large subunit, the gene is located in the chloroplast DNA. But for the small subunit, it is located in the nucleus. It will be expressed into protein, and then with the help of chloroplast transporter, it will be entering into the stromal components of the chloroplast. Okay. Now more to go about chloroplast DNA. It is saying that uh, many chloroplast DNA have two inverted repeats, and these inverted repeats vary widely in length, ranging from 4,000 to 25,000 base pair long each. And these inverted repeats usually contain 3 ribosomal RNA and 2 tRNA coding genes, but they can be expanded or reduced to contain as few as 4 or as many as over 150 genes with the help of different modification of the genes. So most of the chloroplast DNA contain 2 inverted repeats, and the length of these inverted repeats vary from 4,000 base pair to 25,000 base pair. So it varies. It is not similar in size, and within these inverted repeats, it contains regions that code for 3 ribosomal RNA and 2 different tRNA coding genes. But they can be expanded or they can be reduced to as few as 4 or as many as over 150 genes. So within these inverted repeats, it may contain so small amount of tRNA and rRNA, or it may contain as many as 150 different genes within the repeats. Now these inverted repeat regions are conserved among many land plants and accumulate few mutations. Means it does not vary among the land plants. The inverted repeat sequence are same among the land plants. It is conserved. That means it will not change too much. That's why very few mutation occur. Very few change in their base sequence occur within the inverted repeats. Similar inverted repeats exist in the genome of cyanobacteria. Now, same type of inverted repeats have also been observed within the cyanobacteria. It is possible that the inverted repeats helps to stabilize the rest of the chloroplast genome, as chloroplast DNA have lost some of the inverted repeats segments to get rearranged more. So same type of repeated sequence are found within the cyanobacteria, the blue-green algae, and it is possible that this inverted repeats helps to stabilize the rest of the chloroplast genome, as chloroplast DNA lost some of the inverted repeats segments which tend to be rearranged more and more. Now, in the next point, it has been mentioned here that chloroplast DNA have long been thought to have a circular structure, just like mitochondrial DNA. Scientists thought that chloroplast DNA are also circular, but there are some evidence which suggests that chloroplast DNA are takes in the form of linear shape. Over 95% of the chloroplast DNA in corn chloroplast have been observed to be found in branch linear form from rather than individual circles. So just like nuclear DNA, chloroplast DNA are found to be circular in nature. New chloroplast may contain up to 100 copies of their DNA, though the number of chloroplast DNA copies decrease to about 15 to 20 as the chloroplast age. So it's just like mitochondria, up to 100 copies of chloroplast DNA can be observed within the chloroplast genome, found within the chloroplast organelle, but it may decrease as the chloroplast ages. 15 to 20 copies of chloroplast DNA may be found in case of old chloroplast. They also packed into nucleoids just like mitochondrial DNA by associating with the proteins which can contain several identical chloroplast DNA rings. Many nucleoids can be found in each chloroplast. Though chloroplast DNA is not associated with it with true histones, in red algae, a histone-like chloroplast protein called HC protein coded by the chloroplast DNA, which is tightly packs each chloroplast DNA ring into a nucleoid that has been found. So, though chloroplast DNA is not associated with true histones, that's why we term them as nucleoid. But we have seen in red algae that a histone-like protein, okay, that protein is coded by the chloroplast DNA and it tightly packs with the chloroplast DNA just like a nucleosome that we see in chromosome. So it's made into evolution, right? In the primitive red algae, the chloroplast DNA nucleoids are clustered in the center of a chloroplast, while in green plants and green algae, the nucleoids are dispersed or spreaded throughout the stroma. So in the primitive or prehistoric red algae, chloroplast DNA nucleoids are clustered in center, means they are aggregated to a center. But nowadays the green plants and the green algae, they have nucleoids spreaded throughout the stroma of the chloroplast. Now this is a typical diagram of a chloroplast DNA. I have said that chloroplast DNA are linear in nature, but I have seen here circular structure. So here we can see that all the four different complex, okay, which has been shown in different colors. Like you can see here, if I mention one by one, where are the ribosomal RNA genes are located? Here you can see the small rRNA and the large rRNA is coded by the uh, here in these regions. Okay, well you can also see some tRNA coding genes, tRNA for valine, tRNA for phenylalanine, tRNA for proline, tRNA for threonine, tRNA for glutamine, tRNA for leucine, tRNA for serine, tRNA for histidine, tRNA for arginine, tRNA for glycine, tRNA for lysine, tRNA for leucine, tRNA for serine, and all the other tRNA encoding genes are observed here. Well, you can also see the red one, which are the transfer RNA genes that I am just talking with you now. Well, the red ones are the genes that will encode for the tRNA regions, as you can see here, as you can see here, here, it is dispersed, right? It is dispersed throughout, okay, throughout the genome. Then comes the complex one, which is NADH dehydrogenase genes. These are found scattered on most of the regions of chloroplast DNA. Then comes the complex 2-3, which is coenzyme Q cytochrome C reductase genes. Well, they are shown in individual colors. So just go through this diagram and just what I have studied, I have taught with you today. Okay, just go through it. Okay, you can also see cytochrome B here written here. That's the point. That's the point. Cytochrome B is written. So that's all for today's lecture. Uh, go through these two videos that I have sharing to you now. Okay. For any questions regarding this chapter, please ask me in the WhatsApp group. Okay.